Okay, I think we can start. Yeah.
Yeah.
Okay. So welcome everyone to the monthly seminar series organized by this ESAP-funded special topic network called Integration of Speciation Research. So you can find more information on this network on its website, and from there people can also sign up for to the network mailing list to receive automatically the program of the seminar series and other news. So today we will be starting a new series of 3 seminar sessions running in April. So today, May and June before a summer break. And we will be addressing a new general topic of how to understand the relative importance of different biases to gene flow, and also how to outline best practices to measure them with a focus on phenotypes and traits. And in this first session on this topic today, we will more specifically tackle the question of what is reproductive isolation and promises and pitfalls in measuring reproductive bias, with Anja Westram and Daniel Ortiz-Barrientos as speakers. So before I introduce our first speaker, let me remind you how the session will be organized. So each speaker will talk for about 20 minutes, leaving a few minutes for each talk, after each talk, for specific questions to each speaker. And then we will start a more general discussion session, staying on the same webinar link. And so if you have any questions or comments after the talks or during the discussion session, please write them in the chat and not in the Q&A if possible. And we will talk them through during the sessions. And so it's now our pleasure, great pleasure, to welcome Anja Westram as our first speaker. So Anja is currently a guest professor at Nord University in Norway, and also still part-time at the Institute of Science and Technology in Austria. So she obtained her PhD from EWAG ETH Zurich in Switzerland, before moving as a postdoc to the University of Sheffield in the UK to work in Roger Butlin's group. And then a few years later, to IST in Austria to work in Nick Barton's group. So her work on adaptation and speciation combines field surveys, morphological and behavioral phenotyping, as well as genomic and theoretical approaches, with a specific focus on the marine snail Litorina saxatilis as a model system. And so today, Anja will give her views on the topics session on what is reproductive isolation. So please, Anja, whenever you are ready, you can share your screen, and we look forward to listening to you.
Thanks a lot for the nice introduction, and also thank you very much for the invitation. I've really enjoyed the previous session, so I'm really happy to be here and also really looking forward to the discussion, and we think it will be controversial. I hope you can see my screen and
yes,
can hear me well. Yes. So what is reproductive isolation? Before I start, I would first like to acknowledge my amazing collaborators that have been working with me on reproductive isolation. So I think all of them are actually also here today. So this is Sean, Parvati, and Nick. And so we kind of, in our discussions, we noticed that reproductive isolation, the term is used a lot, of course, in the speciation literature, but it's almost never clearly defined. There are a lot of papers that use it and kind of assume we know what it is, and the definition isn't written anywhere. And so in this paper, we try to discuss a bit how to define RI and which definition we think is the best. And we also try to discuss how to quantify RI and how to measure it in practice and whether that is possible at all. And so just to make some little advertisement. So this paper has recently been accepted, so it's not out yet, but stay tuned for that one. So this is what I will talk about in the first part of my talk, and then in the second part I will talk a bit more about my own recent thoughts about reproductive isolation. So that part is not approved by the others, so you can't blame them if I say something silly there. So yeah, if we look at the literature or talk to people, I think there are, in very general terms, two ways how reproductive isolation is discussed. So one is more a focus on specific barriers or on differences between populations that then lead to some kind of barrier to reproduction. For example, we can have differences between populations that lead to assortative mating and therefore to prezygotic isolation, or we can have differences that lead to some incompatibility in hybrids and so lead to postzygotic isolation. So this view is focused on trait differences between populations. And then the other view on reproductive isolation is more equating it to the degree of speciation that a population pair has reached. And this focuses on this continuum from a panmictic population to completely isolated species between which no gene flow is possible. And RI goes from 0 to 1 along this continuum. So this view is focused on the extent of gene flow between populations. And so if we try to put these views together, we can come up with a verbal definition of reproductive isolation. And this definition that we use in our paper is that reproductive isolation reflects the extent to which genetically based differences between populations reduce gene flow between the populations. And because this is a bit long, and it's also very important, I will repeat this with this lizard example. So imagine we have two populations of lizards. There is some gene flow between them, but because there are genetic differences between the populations, they have some differences in some traits. So here illustrated by the different colors. And those genetically based differences, they effectively reduce the gene flow between the populations. For example, because hybrids are less fit, or because these individuals of the different colors don't like to mate with each other. And so if we have this verbal definition, based on that, we can come up with a quantitative definition. So this is the only equation that I will show. And so if we want to quantify RI based on this definition, what we need to do is basically we need to compare this gene flow that is reduced due to genetic differences between populations to the gene flow we would expect if there were no such genetic differences reducing gene flow. So then we get this very simple equation. And I will also illustrate this with the lizards. So again, we have our two populations. We have some migration rate between the populations, and that is denoted M, as usual in the population genetic literature. But then, because we have some differences between the populations, actually a lot of these migrants, they will never successfully reproduce. They will, for example, die in the recipient population, or they will not find a mate. And so this effect of a reduced fitness of immigrants, for example, this can mathematically be simply expressed as a migration rate that is reduced in the first place. So mathematically, the hybrids or immigrants not being successful is equivalent to saying the migration rate was lower in the first place. So we have an effective migration rate that is lower than the normal migration rate, we can say. And so we get the simple measure of RI, which relates these two to each other, these two different migration rates. And RI goes from 0 when the effective migration rate and the actual migration rate are exactly the same. So when there is no isolation between the populations, and the RI goes up to 1 if effective migration between populations is completely absent between the populations because of the genetic differences. So RI is 1 if they are completely isolated, which is what we would want from a measure of RI. And something important to note here is with this measure is that we can see this reduced effective migration rate as a reduced effective number of individuals migrating, or we can see it as a reduced effective number of alleles migrating. So we can have both an organismal and a genetic view on this equation, and those are both fine. But we need to consider that those will be the same as long as we are thinking about an unlinked neutral locus that is not affected by linked selection or assortative mating or something like that. But if we think about a locus that is involved in assortative mating or adaptation, then reproductive isolation at that or around that locus will go up, because of course those loci experience a more reduced effective migration rate compared to the rest of the genome. So RI varies across the genome, and if we want a number of RI for the whole system, we need to think about an unlinked neutral locus. So now we have this definition of RI, and of course then we want to know how does this actually relate to what we study when we study reproductive isolation empirically. So I'm here going through a few examples of how people often study reproductive isolation and relate those to this measure. So first, as I've mentioned already, we have experimental approaches where we, for example, do bait choice experiments with individuals from different populations, or we do crosses and look how fit the hybrids are, for example. And so this, of course, doesn't directly measure a migration rate or an effective migration rate, but it does give some idea about the genetic exchange that is possible between the populations. So to some extent, this reflects RI. But one important thing to note here is, of course, that this approach usually doesn't consider all the barriers. So for example, if we do a matrix experiment, of course, we're not looking at post-zygotic isolation. So these kind of experiments, they look at components of reproductive isolation. And also something very important that is sometimes forgotten is that with this approach, we're typically only looking at a limited number of generations. So if we want to study gene flow, this is, of course, it's a rate over longer times. And if we do crosses, we typically just look at a couple of generations. And so these differences can to lead very different conclusions. For example, if we do a cross, it might well be that the F1 hybrids are very fit, but then the F2 or F3 would break down. So this can lead to discrepancies between these experimental approaches and what we think RI actually should be. And then a different way of studying RI is, of course, with genomic data. So calculating FST either for the genome as a whole or doing an FST scan along the genome. And of course, it is known that FST to some extent reflects the effective migration rate between populations. So this approach isn't completely unreasonable, but we also know, especially from the recent literature, that FST is also affected by lots of other things. So FST certainly should reflect RI to some extent, but a lot of other stuff goes into it. So it has to be interpreted with a lot of caution. And something else that is really important to note here is that because if there are barriers to gene flow, always the whole genome will be affected by them to some extent. So if we measure FST, we never measure the migration rate. We always measure some effective migration rate. So if we think in terms of this equation, we always get the ME, but we never get the M. So we would need to get M from some different approach, which is actually quite tricky. So FST basically measures just a part of this equation. And then the third approach I would like to mention is hybrid zone analysis. So there we're not just comparing two populations, but we're taking a spatial sample from one population to the other, and we're following allele frequency changes or trait changes in space. And then we can do Klein analysis, where the shape of the Klein, the steepness of the Klein gives an indication of the strength of the barrier to gene flow. And of course, this is also not directly equivalent to this measure of RI, because now we're looking at continuous space rather than at this 2D situation that I have discussed for this idea of RI. But still, this is relatively similar in concept to this measure of RI that I have defined. And the difference is just that it works in continuous space. And in fact, we think line analysis is one of the most promising ways to study RI, because it doesn't suffer so much from these disadvantages that I have mentioned for other types of methods. So now in this first part, let's see how long. Yeah, I'm fast, that's good. In this first part, we have seen that we can come up with some definition of RI, and of course, I'm interested to see if other people agree with this definition. And but we have also seen that all the things we measure, they are kind of related to this idea of RI, but they don't directly measure it. And actually, it's very hard to come up with something that exactly measures this. So there is already some trouble, but there's also hope that these give us at least some idea. And so now in the second part, I would like to talk about some more general problem that I think we have with our concepts of RI, which is somewhat independent of the exact definition that we use. And this is what I call the problem of space. And as you might have noticed already in most of what I have said, I've basically assumed we have two nice teams and there is some migration between them, and we are in this very simple spatial situation. But of course, the reality is not like that, right? In reality, we sample the two populations. They are somewhat spatially separate, but there is something between them. There will be some individuals between them, at least in most cases. And those individuals basically connect our focal populations like some kind of corridor across which gene flow happens. And then the question is, when we think about RI, do we include this corridor in our thinking or do we not? And based on this, I believe that there are two different ways of looking at RI. One of them I call pairwise RI, and that is if we really think about RI just in terms of those two focal populations. So we have the red and the blue population, we put them together and we ask how much gene flow would there be between them? And this is hypothetical gene flow, usually because there's something in between that we are ignoring. They are not directly connected in nature. And the other way of thinking about RI that I will talk about is this I call context RI, and there we're actually considering that there is this corridor and we're considering the gene flow across this connection in nature. And I mean, maybe you're looking at this now and thinking, okay, one of them is silly, it's obvious which one to choose, but I would just like to highlight that with the methods that we are using to study RI, we are applying both of these ideas. We're just often not explicitly making this distinction. So for example, if we do crosses that I have already mentioned, we typically take individuals from this population, from that population, put them together and see what happens. So we're kind of ignoring that there are individuals in between. And so we have this pairwise idea of thinking about RI. But on the other hand, if we do something like an FST scan, okay, we're also taking individuals maybe only from here and here and not individuals in between. But of course, the FST we get, it depends on the connection between them. It depends on the gene flow that happens between them and what kind of individuals or genotypes are there. So this is a way of measuring context RI. And so to illustrate that these really make a difference, so it really makes a difference how we think about RI, I will show you two little examples. So here again, we have space. And in this case, we have a red and a blue population, but we assume that the red and the blue allele, they are incompatible. So if they come together in the same individual, the individual will most likely die and not reproduce. But so in this example, these red and blue alleles actually never meet in space, because here we see the clines of the two alleles and we see that the blue allele, it goes down in frequency before the red allele goes up in frequency. So in nature, they never meet. And this kind of displacement of clines that is predicted by theory and has also been observed in empirical studies. So this is probably not some strange extreme case. And so if we measure RI in this situation and we apply this pairwise thinking, then we would take individuals from here, we put them together, maybe in a cross, and we would see, okay, they almost never reproduce because all the offspring die, or I mean, they do reproduce in my example, but the offspring will die before they can reproduce. So we would think, okay, there's a strong incompatibility and RI must be really high. But then if we go to the field and we measure the gene flow between these two populations somehow, then gene flow is almost not impeded because the incompatibility is never expressed. So our context RI will actually be quite low. So we get very different results from these different ways of thinking about RI. And then here I have another example which gives kind of the opposite result. So here we have two blue populations that we are focusing on, but between them we have an area of red individuals. And in this example, we actually have expressed incompatibilities in the field. So here we have the clines of the red and blue alleles basically colliding. So here in these two areas, where the red and blue come together, we have very strong barriers to gene flow. So it will be very hard for an allele to cross this boundary or this boundary. So in this context, if we again do the experiment, we put these and these individuals together, we will of course think RI is very low because these populations, they are genetically very similar. There are no incompatibilities between them, so we will get a low estimate of RI. But if we check what is actually the gene flow in the field, this gene flow will be quite low because the genes have to cross these two barriers in space, and we will get a very high estimate of RI. So again, we get completely different pictures by these two different ways of thinking about RI. And so, okay, now the question is, of course, which one is better? Is one of them stupid and we should forget about it, or are they maybe both useful? And so before I try to answer that question, I will just quickly repeat what the difference is between these two ways of thinking about RI, just to really make that clear. So if we think about pairwise RI, this is really a property of a population pair and nothing else. And what we are asking when applying that way of thinking about RI is we're asking to what extent are there segregating barriers or potential barriers between the populations, because they are not really expressed in the field. We might only see them in the lab. And on the other hand, if we think about context RI, this is not really just a property of a population pair. This is more something like the status of the population pair within the system in which it is embedded. And I think here a useful thing, at least to some extent, is to think about reproductive isolation as somewhat analogous to geographical isolation. So here populations can basically be geographically distant, but we can also say they are reproductively distant. So for example, in this example, we could say having this red area here or having these boundaries between the two areas here is like adding more geographical distance between these two focal populations. So this kind of thinking is more similar to this way we think about barriers in hybrid zones, where we can express the barrier to gene flow as a geographical distance in space. So there is some similarity between reproductive isolation and geographical isolation, but they are of course not the same. So here we are more asking to what extent are the populations evolving independently because they are separated by some form of distance. So which one is better? I would say they are both important. I would say we have to really look at both of them to understand the speciation process. So if we are only looking at segregating barriers, we are not really studying the extent of speciation because maybe the barriers aren't expressed, as we have seen in the first example. But if we only look about the extent to which populations are independent, are evolving independently, we are also not really measuring the degree of speciation properly because they might not have any genetic differences as we have seen in the second example. So I think both of these in some sense signify closeness to speciation, but neither of them alone does. So I really think we have to look at both of them together. And we also have to be aware that we have these two different ways of thinking about RI, and we need to be more clear about specifying which one we are thinking about when we are measuring something in our analysis. And I would really like to highlight that I think, except in very simple cases, of course, we can really not describe reproductive isolation with a single number. So we can, to conclude, we can define RI hopefully in a way that is consistent, but it's very difficult to measure in practice. And there are these two different interpretations of reproductive isolation. And I would say that we really need to consider both, and we need to be clearer about the fact that we do, and also we need to be more specific when interpreting our data about what we exactly mean by RI. And with this, I hope I haven't taken too long. I would like to again thank the people I have worked with on reproductive isolation. And I would also specifically like to thank Roger and the Barton Group for a lot of interesting discussions. And finally, I would of course like to thank the audience for your attention and the organizers again for inviting me today. Thank you.
Thank you very much, Anja. Very interesting perspective on this question, difficult question. So if you have any questions for Anja, please write them in the chat. I don't think I see any right now. Okay, so the first question from Jona. So could you elaborate how the context RI can be used in systems that don't have the traditional hybrid zone, like a mosaic hybrid zone we find in the woodlands?
Yes, I mean, that's a good question, because I mean, I haven't talked about how to do this in practice. And I mean, there is a good reason for that, because I personally think it's really difficult to measure RI. I mean, it's, of course, we can do crosses, and that's probably part of the reason people do crosses, but really measuring what is going on in the field, I think it is very hard, right? So I don't think I have a particularly good answer to that, because I think we can get some idea about these things by doing genetic studies that are more spatially uninformed, maybe I would say. So I think something that I would find important is to, I mean, I think I didn't talk about that very much, but I think in general, it would be important to really consider speciation in space and to be less biased when we do the sampling, right? So I think it would make sense to have projects where we really go out and sample in an unbiased way and look at genetic structure without predefining populations by nice traits that we are interested in, for example. And I think, I mean, this is also not just my new idea. Of course, a lot of people promote that view, and I think Roger, for example, has also said for a long time that we should maybe not just sample those individuals that are interesting because they have different colors, but have a more spatially continuous sampling without any bias. And so, I mean, one way then to look at it is, of course, looking at genetic structure, but I also already said in the talk, and we all know that the genetic structure doesn't really reflect RI, especially not fully, and especially not in complex systems. And I think then it's really hard to know how to then look at reproductive isolation in these systems. So yeah, I'm not sure I can say anything very intelligent right now.
Well, thanks. That's great. Maybe one question from Xavier Vecmans. So he says that you presented 3 methods to measure RI. But what about inference and speciation model testing using ABC or other methods? Don't you think that estimating the proportion of the genome that fits an IM model versus strict isolation model is not a way to estimate RI at the genomic level?
Well, I think, again, I would say that maybe I was a bit negative, right? I think it's not so that these measures are not telling us anything about RI, right? I think to some extent, yes. But I also believe that this kind of analysis has a similar problem as studying FST, for example, because if we do some analysis using ABC or using any kind of demographic modeling approach, this estimate of M that we will get will be an estimate of ME, because if we have barriers to gene flow that affect the whole genome, we cannot estimate M from the genomic data because the whole genome is affected. So I think with genomic data alone, used in demographic modeling or used in an FST analysis, we never get around the fact that we don't have an estimate of M from that data. And I think, of course, we can in principle get estimates of M from other sources, like from some mark recapture experiment or something like that. But that is also, that's of course problematic to compare a measure of ME, which we have from genomic data, which reflects a lot of generations, with some measure of M that we have from the field, from a mark recapture experiment that we did over a year or something like that. So yeah, I think it's still kind of tricky.
Yeah, so other people mentioned that, like the difficulty of measuring M as it relates to potential migration in the absence of the barriers. That's a comment from Mathieu Joron, for example. And Daniel also is asking what you think about the error around the estimates of RI given your formula. I mean, it's practical issues, I guess, on how to measure this.
Yes, I mean,
ratio.
Sorry.
Yeah.
I think it's. Of course, I would like to hear what other people think, because I have become very negative about measuring RI, right? I think everything that we do, it can tell us something, but it can give us a feeling about RI. But I'm not sure we can really get nice numbers. I also by now think this equation that we have, it's not, to me, it's not something that I would actually use a lot to measure RI and get a number. It's just to have something that we can write down and agree on, that we think this is what reproductive isolation is. I'm not sure my co-authors agree about this, but I think I would think it's very rare that we can actually get some nice number from this, and especially that we can compare that number between different systems and really be comparative in a quantitative way.
Yes. So we have a few more questions, but maybe we can go back if we have time after Daniel's talk. So we should now invite Daniel.
Yeah, so it's my great pleasure to introduce Daniel Ortiz-Barrientos to give the second talk today. Daniel started off doing a PhD with Mohammed Noor, working on Drosophila pseudoobscura and persimilis, and looking at multiple components of reproductive isolation in that system, and particularly at the genetics of reinforcement, where he revealed a really interesting potential one allele example, a rather rare example, it has to be said. He then worked for a while on human genetics and on sunflower speciation, but he's currently at the University of Queensland in Brisbane, where he's been for quite some years now developing a really interesting system in Senecio lautus, which gives beautiful examples of repeated differentiation and the appearance of different forms of reproductive isolation. So we're very interested to hear his alternative view on what is reproductive isolation. Thank you, Daniel. Please take it away.
Thank you very much. Thank you very much for having me. It's truly a pleasure. So I'm going to share my screen first and make sure that we are all on the same page. So can you please confirm that you can see the full screen?
Perfect, yes.
Okay, wonderful. So hi, everyone. So as I said before, I'm incredibly happy to be here with Anja, sharing with you some of our ideas. And I want to thank the organizers for the invitation, Carol, Roger, Stan, Joanna, Elizabeth, Jonah, Christopher, and the rest of their wonderful team. Thank you so much. And I hope we can have more of these initiatives down the line. I think they are terrific. So today what I want to do is to argue for 3 ideas that might generate discussion, right? So the first one is that there are multiple types of species. The second one is that we need to focus our efforts in studying trait evolution. And the third one is that we're not actually interested in the species problem, but in the discontinuity problem, right? So I will act as a defender of them. but I hope you can push back or join me in defending them. And I think that will just be an exercise in helping us think more careful about the nature of the species, their origin, and more broadly, the origin and maintenance of biodiversity, right? Okay, so let's start with a very simple example of reproductive isolation that we're all know, like the existence of mules. And as you know, these mules are sterile offspring from crosses between donkeys and horses. And like them, there are hundreds of examples in which crossing two divergent taxa leads to sterile or dead offspring. Now, such effects of hybrid fitness manifest in any type of environment. And we refer to this process as intrinsic reproductive isolation, and it works. In this extreme example, there is no recombination of genetic material between the two groups, the one in the left, group 1 black, and the one on the right, group 2 blue, right? C'est fini, that's it, like there's no more. Now, we can also have partial sterility in the two groups, or only a fraction of individuals dies, right? In any case, this works because intercrossing will recreate hybrid allelic combinations that reduce fitness, and will also recreate parental combinations that remain fit. So overall, this will be also an effective way to keep the two populations apart, right? Now, populations might be viable and fertile in laboratory conditions, but sometimes not under natural conditions or under severe stress, right? So in this example, we have two forms of fish, and they were crossed to produce offspring with different amounts of genome from each form. So on the left, where you are examining fitness, you see an increase in the proportion of local alleles, percentage of genome form A, increases fitness of those individuals in that environment A. And the same happens in habitat B. Individuals with more portion of the genome derived from local individuals will have higher fitness. This is, the way I see it, is just the typical signature of adapted forms, and it can create reproductive isolation where the forms meet, right? Now, I think that despite our tradition of categorizing reproductive barriers in multiple types, and Anja alluded to this very nicely, I think that, there are two questions that we normally have in mind when understanding reproductive isolations. A when question, which is basically a developmental question, right? Where is the development diverging, right? Where is the development becoming impossible to overlap between two groups, right? And this will contribute, as Anja was saying before, like to that discrepancy between ME and general M. And the second one is a where question. We have a genetic component, but the genetic component might actually differ across environments, right? And it could be across specific environments where the organisms occur, or just across any environment that is stressful, okay? So I like to suggest a way to look at reproductive isolation is through this equality, right? So we have on the left, a population P1 that is evolving traits that have a genetic basis, G, but those traits might or might not interact with different environments. And you're looking at those traits at different developmental stages, represented here by the D. So with this in mind, I think we can say that reproductive isolation answers this null hypothesis that the two populations remain evolutionarily sort of exchangeable. You can actually transfer individuals from one another in any way, and you cannot reject this equality. Now, this is of course a relative concept, right? So, and this is important because speciation happens in the equal sign. And the thing that happens within P1 or P2 is actually trait evolution, right? So the way to study speciation is one way in which we need to focus a lot on what happens within populations, but also test against what is happening in other populations. And it's that relativity that brings us to study speciation in a powerful way. So we can consider trait evolution happening by default, because mutation, drift, and selection will basically ultimately reject this equality, right? But there are ways in which we can actually maintain it, right? So, or with great difficulty reject it, right? So a power issue as well. So on one hand, we have young systems, massive levels of ancestral polymorphism, and rejecting this equality is really tough. And it's going to be a technical issue to actually do it, although there might be examples in which very few genes might drive the evolution of a very powerful trait, like let's say things that that are related to color. We know that they can have major impact on exchangeability of this information. And therefore, there can be young systems with loads of ancestral polymorphism that still can reject this equality quite easily. Now, another way to keep it or to be unable to reject it is a lot of gene flow, right? And you can recombine the two genomes all the time, and it's very hard for these traits to evolve from one another. Now, we can reject it. We can just remove recombination altogether, right? And here's where traditionally we have thought about allopatric speciation, right? And as Anja mentioned, I mean, this is a simple case in which spatially the two groups are fully apart, and in a pairwise context, I would say that there are no intermediates. They're truly allopatric, right? But the reality is that we have a continuum in many occasions. There can be more complex ways to remove recombination. They can also be based on mechanisms that suppress recombination in specific areas of the genome, like as we know, the effect of chromosomal inversions in keeping swaths of alleles that are adaptive from recombining and creating maladaptive combinations, or just locking into place genetic incompatibilities. So overall, we have, on one hand, trait evolution. On the other hand, we have recombination. And the geography seems to matter a lot. So in allopatric populations, apart to one another, I don't think that we're studying a speciation per se from the point of view of reproductive isolation. We have dealt with it by adding potentially reproductive to the definition of biological species concept, but it's definitely an addition. And it's an addition that allows us, it's very important though, and it will allow us to do some things that I'll talk about in a second. But overall, honestly, what we're truly studying and is happening in those populations is trait evolution. But in places where populations come in contact or are in contact or are sympatric, then there is an actual means by which reproductive isolation counts. And I would argue that recombination, in that sense, or places where recombination matters, is where we can truly understand the genetics and ecology, etc., of a speciation. So, expanding on these ideas, we have for allopatric populations and trait evolution, we can think that, yeah, we're studying allopatric speciation, but it seems that on a kind of pushing the envelope here, we are studying allopatric speciation, but that is basically understanding lineage sorting. And there are many drivers of that type of speciation. And they are great to test the null hypothesis, but that is just to get a feeling for how the genetics, let's say, of the traits, developmental traits that would potentially create reproductive isolation are evolving. And it gives us a reference to compare it against the genetics to when the populations are recombining with one another. Now, in these populations, we can definitely study genetics of trait differences. It's wonderful for that. And again, if you study reproductive isolation here, yeah, you can sort of like try to find possible rules as to how trait evolution itself could potentially create reproductive isolation, right? And in terms of species, well, I mean, I think that molecular systematics might be a really good place to define and to actually make coalesce our day-to-day definitions of a species with historical divisions of groups, right? Now, with recombination, when we have sympatric and parapatric cases, right, we're actually studying natural means by which reproductive isolation evolves. And we are actually testing the hypothesis, not experimentally, although it can be experimental as well, of course, but it's being resolved by natural experiments. And that's a great distinction between the two. And here we can actually study the genetics of traits as well, but we can link it to the genetics of a speciation. And here where biological species or phenotypic and genotypic clusters can be easily used. One thing about genotypic clusters is that they can also be used in allopatry, but then it's harder to know when they actually are biologically meaningful. And it's going to be always the case when we have form or a metric that is continuous, right? Now, so to some extent, I guess we can, I told you that I was going to try to be controversial here. We can have trait speciation and recombination speciation, right? And both are types of speciation, and it just both contribute to the origin of this continuous diversity, right? So we can study both, and we are with both addressing the problem of discontinuity, organic discontinuity, right? And I would say that by the nature of the genetics of the two processes, the ecology, the geography, and how these forces interact, they are effectively two types of diversities. And it would be great to know what are the true commonalities, and if the true commonalities actually matter, how they bring together the two processes beyond same potentially, and how we can actually study them in actuality. And for that, I actually come together with Anja in saying that secondary contact might be one of the most interesting cases where we can actually test the two modes coming in one single place. And the genetics of allopatry, comparing it to genetics of sympatric or Klein's, etc., as I show here, they are the ones that might actually help us understand the connection between trait evolution and recombination, and trait evolution in the presence of recombination. So I think that secondary contacts are very interesting in and of themselves, but actually are a natural experiment to test the biological basis of what I've been calling like trait speciation and recombination speciation, right? So I've been traditionally a geneticist, so I think that we need to focus a lot on the genetic architecture, but it's not enough. I think that we need to bring into bear the ecology and the geography, of course, to get a great picture of the process. So what now? So in my humble opinion, a student of speciation is a student of organic discontinuity, right? That is fundamentally interested in trait evolution, and that is studying fundamentally the interplay between evolutionary forces like selection and recombination, right? And that is interested in all the causes of diversity, not only one type or the other, but actually trying to test at its core how that discontinuity actually takes place. Now, as a student of speciation, we conduct lab experiments and natural experiments, and we take data from both. And echoing like what Anja was saying, in context of speciation, well, I mean, we should definitely sample broadly, and not only in a pairwise matter, but actually try to encompass much of what we can across the range of a particular group. And ideally, we want to integrate genetics, ecology, development, and by genetics, I mean, very broadly speaking, including PopGen, population genomics, and so on and so forth, right? So this is more or less what we try to do in my laboratory. And as Roger mentioned, this is the Senecio Lautus species complex. It's highly diverse, and it's basically a system that has allowed us to think about this type of framework. And I'm gonna just briefly show you a couple of things that a student of mine, Henry Arenas Castro, has been basically doing during his PhD. And he's been basically focusing on the evolution of gametic interactions. And he has been basically following a comparative experimental study, right? And in this approach, basically, we're trying to look at the combined effect of time, ecology, geography, genetics, and barriers all in one, in a single experimental design. So with this in mind, we want to actually test many of the aspects of this equality. So we have systems in which we can sample across the continental range, in this case of the species complex, and we can vary geographic sampling, sympatric, allopatric, or parapatric. We can also infer through molecular methods the relatedness of these populations and take that into account, and track the ecology of the system and other aspects incorporated in the analysis. So ideally, we create experimental designs that here on the left, you see the variable is basically the test of the hypothesis of equality, and we try to see what is driving that equality. And experimental design incorporates all of that. It's a single experiment, and we hypothesize the different forces that could be driving it, and how that might speak about the speciation. Right? It's an very simple one, in which here in the upper diagonal of this correlation of this matrix, you have a competitive gametic isolation. Pink is strong and blue is weak. And on the lower triangle of the matrix, it's non-competitive isolation. And what he's found is that non-competitive isolation is weak. In other words, you just exchange pollen between the populations and you don't see much isolation at all. But if you compete them, then it becomes stronger. So this idea that plants might be very fertile, well, I mean, maybe one at a time. But when they meet in areas of parapatry, for example, it's unlikely that that is the only way in which these populations are being isolated. And a second example is how ecology matters here. So on the top, you have all the dunes, which live in sand dunes. And on the bottom, you have headland populations which live in rocky outcrops by the coast. And in general, the message is that biases in fertilization are highly dependent on the ecology of the dam, not the sire. Sire could be anyone, but the mom has to be the actual mom of the place, and they are super picky against anybody else, right? So with all of this, what we want is just to answer many questions, right? Which this basically just touch more upon the way we conceptualize it at the moment, which is what do patterns of reproductive isolation tell us about the speciation process? I think that the idea of what reproductive isolation is intimately related to what it can tell us about the speciation process. And in some cases, it might not tell us anything. Also, it helps us study the traits that drive those patterns and the degree of discontinuity that we see across the system. And that discontinuity can be partitioned by geography, ecology, and so on and so forth. And in a given system, you might have dynamics that are completely different from one another. In other words, you might have purely allopatric populations, and in other cases, you might have purely parapatric ones. So we need to consider that when generalizing our views about discontinuity. One important issue is the genetics of barriers. I mean, I think that we don't know much about how pleiotropic they are and how correlated they are. And I think that there is an aspect that is very important and that might differ quite a lot based on data so far that we have, the little data we have, might differ quite a lot between allopatric and parapatric cases, for example, or trait speciation and recombination speciation. And finally, with that, we can all together kind of like bridge the gap of finding the evolutionary forces there. So this is a lot of the people in my lab and kids, and we are all together kind of like working on these aspects from different angles, and many people working on the trait evolution approach and working on adaptation per se, others on the interaction between populations at different stages of development, and also per se like the evolution of recombination. So we can actually study it in the different geographic settings, for example, right? And always interested in people joining the lab, so just hit me a note if you're interested. And with that one, I'm just going to say thanks to the organizers again, and to the audience for coming, and for Anja to join efforts in creating this discussion forum. And I leave the 3 points that I made at the beginning there, just for us to keep in mind. Thank you very much.
Thank you very much, Daniel. That was fascinating and interesting contrast, some interesting contrast with Anja's view there. So we have a few minutes for more specific questions, and then we'll move progressively towards the general discussion. One question came up in the chat during your talk, which was about ecotypes. Alexandra Weber asks if for ecotypes, the equality you use is surely not true. Does this mean that ecotypes are equivalent to species?
Right, so here I'm going to be incredibly Darwinian in that answer. And I'm going to say that I don't care. and to some extent, I mean, I'm going to give you the two answers. On one hand, we can say that it's completely arbitrary to call them one way or another if there's no recombination at stake. And at that point, we just need to come to grips as to how to define this species. And it could be just in terms of multivariate space, right? Or it could be in terms of like more like the genic view from Chang and Wu, and just trying to find those highly strong effects that are highly differentiated and that drive most of the differentiation there. But also you can argue that these ecotypes, or I mean, they're just partially reproductively isolated, and we can try to figure out what is contributing to that partial isolation, right? So I think that thinking about what our system is telling us and the diversity that we're finding when we sample like dosing populations and everything against everything, and I mean, it seems that it's very hard to pinpoint what a population might be a species or not a species unless you actually are arguing for trait space or unless you're arguing for zones of contact. So, I mean, that's one thing that I still don't know, basically. And if you ask me, every population in my system would be a species because any is 0, and or lightly, or a little bit. If you do transplant experiments and you calculate other reproductive barriers, like for example, as Henry did, some populations are parapatric. They have a small hybrid zone, but total reproductive isolation is 1, right? But you bring them in the glass house and you can produce F1s easily, no problem. But if you do competitive gametic isolation, it's quite strong, but not enough to keep them apart. So there's nothing that explains by itself why total reproductive isolation is complete. But according to everything that we've talked about and from Anja's talk, every single population in the system would be a species. But I don't know if what that actually tells us.
Yes, so I mean, perhaps we shouldn't get too deeply into species definitions, but
correct.
I mean, I think the underlying point is perhaps that your inequality can be broken, but the measure of reproductive isolation is somehow the measure of the extent to which it's unequal.
Correct. Yeah. In that sense, it's very related to what Anja was talking about.
But the problem then is how to measure that. Or whether it's meaningful to measure that strength, and so how do we go about doing that?
Correct.
Yeah, perhaps we can take that opportunity to move towards some of the more general questions. And I mean, one of our first questions was about whether the problem we have with reproductive isolation is primarily about common language, or to describe, all the common ways of using these various terms we have, barriers, barrier traits, components of reproductive isolation, and so on, or whether the problem is actually more a technical measurement problem. So if we could agree on the definitions, could we make the measurements?
Right. I'll go first.
Yeah, go first, and then we'll let Anja have a say.
Sure, of course. I would say that I don't think it's a language problem. I think that there are biological attributes of populations that make them different in how they diverge, whether when you have a recombination or when you don't have recombination. And I would say that if we lump them in a single one, we're sort of like lumping also perhaps many genetic architectures into one single way to produce the barriers, which ultimately I think have to do with the interaction of within population trait evolution and its consequences for the between, right? So I would say it's not language per se, the actual processes, but I think that agreeing on how to call them, well, it's been a debate for decades. It would be awesome to actually come to a common ground to call all of these things, and then face the technicality with ingenuity, I guess, because it's gonna always be there because the sample size or the unit of replication, what is it? Like populations, pairs? I mean, like it's a very complicated approach, right? So if we have one single pair, we need to ask ourselves, what do we learn from that single pair, right? And how do we actually, what is that we need to do to generalize? I'll stop there.
Yeah, Anja.
Yeah, I mean, I have many thoughts about this. I do agree. I mean, I think to some extent it is about language, just because we often don't say what we mean when we use terms like barrier or reproductive isolation or something like that. And then we think other people they understand it because we all use the terms, but maybe we mean something very different. And so I think that is semantics, and it's kind of boring, but I still think it would make sense to, I mean, this is also kind of what you said, it would be nice to use the same language just so that we don't waste so much time on discussing the language. And I think Roger and Carol, for example, had this paper 2 or 3 years ago, I think, about coupling, where they really defined barriers, barrier effects, barrier loci, and all this. And I think I'm not using that terminology, to be honest, but I think it would be useful if people agreed, because I think barrier is also a word where we're not really, it's not completely clear what we mean by it sometimes. And so yeah, I mean, but I don't know in practice how to get a consensus about word usage in the community. Because there will always be somebody who disagrees, and I think it seems a bit random what kind of word usage spreads in the community and what doesn't. And I also think Carol and Roger actually never really define RI in their paper, if I remember correctly, but they do define other stuff based on RI, and so there is still a challenge. Yeah, but so this was the boring part about semantics. Yeah, but I mean, about the processes, for sure. I mean, I think there is some general agreement about our understanding of speciation. I think we all have some kind of intuition of how things work, and that maybe that intuition isn't actually that different between people. But it's hard to pinpoint this. I think we have some need to categorize things into species and to pinpoint things with a single number. And I think to some extent for speciation, we just have to accept that maybe the intuitive understanding is the best we can get, and we cannot have a number that this is RI and this number will work for every system that we compare.